So, ladies and gentlemen, if you could take your seats, please, that would be great. Please grab your seats. We need to get started for this next session. So it's a great pleasure to introduce the next session, which is looking at neurogastroenterology and Parkinson's disease and unravelling the gut-brain connection. We have a group of excellent speakers, and we're also looking forward to a panel discussion when all the questions will be able to be asked at the end of all of the talks. So it gives me great pleasure to introduce as the first up speaker Professor Carolyn Sue from Neuro. She's an expert in Parkinson's disease. She's a neurologist by background, and she's the Kinghorn Chair and inaugural professor at Neuro. So, Carolyn, it's a great pleasure to have you here. We're looking very forward to your talk on From Gut to Brain, Understanding How the Microbiome Impacts Parkinson's Disease. Thank you.
Well, thanks, Nick, and thanks also to Peter and the organisers for asking me to come and talk to you. I'm actually married to a gastroenterologist, and I hardly ever get to come to gastroenterology conferences. So it's a real pleasure for me to come here, but particularly in this session where we're connecting between the gut and the brain. This is where my new location. I actually worked at another institute before being recruited here, but I was actually an alumni here, so I'm very pleased to be back on this campus. I'm a clinician scientist, and I have my clinics here. I'm not sure at this point it's been having trouble working. So we have clinics on the bottom row of this of Neuro here, which is just up the road, about a 10-minute walk, and I have a laboratory up on the top of the building, which has been very pleasant to move into. Okay. So maybe this. Oh, use the mouse. Great. Okay. Lovely. Okay. Thank you. So this is James Parkinson's, who I've got to say that if he wrote about about the constipation in Parkinson's disease rather than the shaking palsy, I wonder whether I would have been a gastroenterologist instead. He originally talked about this wonderful this disease that is now named for him, and wrote this wonderful essay, which talked about the involuntary movements of Parkinson's disease. And of course, all of us are really well aware of what Parkinson's disease knows. Usually, it's a very common disease. Usually, people have known people in the public arena or have relatives that are affected by this disease because it affects over 200,000 Australians, 10 million people around the world. And James Parkinson's talked about these involuntary movements, which are due to the loss of dopaminergic neurons in the substantia nigra, as you can see here. And I'm not sure if this arrow works, but you can see here on the right a Lewy body sitting in the middle of a dopaminergic cell, which is coloured with melanin, which is the material that gives the substantia nigra its black appearance. So the motor function loss in Parkinson's disease is due to loss of dopaminergic neurons in this area, and the pathological hallmark of this disease is Lewy bodies, which contain alpha-synuclein, the aggregated protein in this disorder. Now, Parkinson's also described the gastrointestinal problems that were in this part of the disease. And you can see here that he writes I think this is page 7 of his essay, that the bowels are affected in this disorder and require medicines of very considerable power to overcome the constipation that is associated with this disorder. And of course, very nicely in the first session by George Rowan and Eunice, very nicely set up and alluded to the problems in the microbiome in Parkinson's disease, where we now know that alpha-synuclein not only accumulates in the neurons, but it also accumulates in the myenteric plexus of the bowel, where aggregates are thought to either enter in through the bowel or the brain. And I noticed in the question is, does it start in the bowel or the brain? There is probably two schools of thought and probably people on both sides where you can get a body first, in other words, the gastrointestinal symptoms starting first in the disease course, or entering in through the biome, through the olfactory tract, which then goes down to midbrain. And of course, there's a connection between the vagus nerve here, which creates this bi-directional communication, which occurs in the the gut-brain axis here. Okay, and of course, and of course, alpha-synuclein is the degenerative protein which actually sets up these positive feedback loops. So there's an accelerating process where you get a transmission of alpha-synuclein aggregates up the vagus nerve from the bowel into the midbrain, or vice versa, and then inflammatory responses are generated either in the CNS or the peripheral nervous system, which then creates these positive feedback loops, which then leads to the progression of Parkinson's disease. Okay, so what I'm going to do is talk to you about our work in the gastrointestinal dysfunction in Parkinson's disease. And really this started off with a PhD student of mine, Michael Lubomsky, who then published a series of papers in our findings when we set about a study in looking at the microbiome in a cohort of patients with Parkinson's disease. And the aim of this study was to investigate the associations between the gut and the microbiome and the motor and the non-motor symptoms of Parkinson's disease, and again to develop predictive models that could guide diagnosis and clinical management of Parkinson's disease, because at the end of the day we're neurologists. Okay, so this study, our pilot study for his PhD project, really involved 103 patients, and we decided to look at use household controls because we wanted to have the same dietary intake over this longitudinal study period of time. What we did during this study was we did some deep clinical phenotyping of the Parkinson's disease patients, and I'll tell you a bit about that in a moment. We studied the microbiome using 6S ribosome sequencing, and we measured their dietary intake with food questionnaires, and then a dietitian who then looked at the macronutrients that they were consuming for us. And so this opportunistic recruitment of household controls with very similar dietary habits, I think, was a key to one of our study designs. Okay, these are some of the clinical phenotyping. I won't go through all of it for you because it basically looks at a whole bunch of clinical rating scales that we neurologists do to define the progression and severity of the disease in Parkinson's disease. But we looked fairly extensively at both motor function, non-motor function, psychological impacts, quality of life, as well as the gastrointestinal components, which we're looking at constipation scales, dyspepsia scales, and gut motility. We also looked at cognition and some pain scores as well. These are the demographics of the cohort that we had. We had 103 patients with Parkinson's disease. Most of them were male. There seems to be a slight male predominance in this disease. Obviously, therefore, the household controls were more female. And we looked at the gastrointestinal symptoms. And really, this table summarises the increase in gastrointestinal symptoms that were in our cohort, with increased scores of constipation and dyspepsia. And this is some of the data that we published showing that indeed, sorry, my mouse doesn't work, so you can see the patients in Parkinson's disease patients in blue, that there was more constipation in Parkinson's disease patients and that they had harder stools on their Bristol stool score. This is the microbiome data where we worked with Andy Holmes from the University of Sydney, and he did all the taxonomy for us, and essentially we found that the composition of the microbiome was different between Parkinson's disease patients and the healthy controls at all levels, including genus and family. So you can see this raw data here. Parkinson's are in green and household controls are in brown. And you can see here the data as we stepped out to order and phylum that there were compositional differences of the microbiome. In fact, we could actually tell who had Parkinson's disease by looking at the microbiome instead of actually looking at the patient. There were compositional levels at differences at the genus, family, and order levels when we compared to the healthy controls. And you can see the fold changes here as demonstrated on these volcano pots. So really looking at major differences in the microbiome. And I've summarised this on this table here, where you can see the altered profile suggested there was underrepresentation of some of the bacteria. I'm going to point out here this Butyrococcus. Basically, reduction in species that reduced the butyrogenic production potential in the gastrointestinal tract, and also there were overrepresentations of bacterial species, Enterobacteria and Lactobacillus were some of these other ones. And these bacteria are known to increase the metabolism of levodopa in the gastrointestinal tract. So here we are, we have patients taking levodopa for treatment of their Parkinson's disease, and the microbiomes or the gut flora go, you beauty, lots of food, love this, we're going to proliferate. And so there are species that actually metabolize not only the dopamine, but also degrade the carbidopa, which actually stops the breakdown of levodopa. So, you can see here you're creating a lovely micro environment for these species to proliferate, given that they're being fed by the patient taking the tablets for their disease. So we found, we worked with a whole bunch of data scientists and started to look at some predictive modelling. And I'm not going to pretend that I'm a data scientist at all, but by using random forest modelling, we found that at the genus level we could actually predict who had Parkinson's disease and who didn't. And then by integrating our nutritional data, and in this case looking at the carbohydrate intake, we could improve the predictive model to an area under the curve, to about 0.74. And we're now building on this model so that we can improve its predictive value even more so. We then proceeded to go on to a longitudinal study, particularly looking at the impact of device-assisted therapies. And you can see here, this was during COVID, I've got to say, so we had, out of the 103, we had 74 complete the 12-month follow-up. Fairly difficult during COVID because they had to hand-deliver their fecal samples to the hospital at a time when we were actually closed down for clinical trials, which was a challenge. And I won't tell you the stories unless away from the lecture theatre, but in private I'll be happy to tell you some of the hilarious ways that we managed to get some of the samples into our lab. Anyway, we measured clinical progression with a number of different rating scales, and then we also measured the non-motor scales and also measured the amount of levodopa that patients were requiring to treat their disease. And of course we did sequential assessments of the microbiome. And at 12 months, we found again there was an increase in overrepresentation of the Lactobacilli family, which were metabolising the levodopa, and also continued underrepresentation of the butyrogenic protective species that we had. Again, it was important to notice that as the disease progresses, there was a continual reduction in those species that were protective of the gastrointestinal tract. Okay, this is how we segregated this. So we categorised patients as being fast progressing by the amount of levodopa that they required to maintain their motor function. And so we had only 34 patients who required increasing doses of levodopa over this time, and there were slower progressing patients, 40 out of this small sample. So just remember, these are small numbers, but again, in collaboration with the data scientists, they managed to, again, feed data into their predictive modelling. They combined central log transformation with a support vector machine model to determine who was progressing faster, and they did, a technique where they left one out, cross-validation, so bootstrapping up the data, and working on the two-stage classification model that we constructed, and then fed the macronucleate data and the microbiota profiles into our model. And again, we found that by using random forest modelling, that using the microbiome data and the nutritional data with this two-stage classification of disease progression, that they were able to develop and build towards a predictive model. And the incorporation of protein intake in their total energy intake was actually improved in terms of disease progression when you built it in and combined it with the microbiodata. So we're working on another study with Nick to help to extend this study out so that we can look at microbiome profiles and data and dietary information, as well as a whole bunch of other parameters which will help us with this predictive model. So in summary, the gut microbiome profiles combined with clinical measures and nutritional intake can help to identify Parkinson's disease patients. Altered gut microbiome profiles in Parkinson's disease can persist, at least for 12 months, and we demonstrated there was always an underrepresentation of protective species and that could lead to the gut leakiness due to these reductions in the butyrogenic production of microbiota producing species, and that the specific gut profiles combined with these dietary factors can help us in the future to predict the progression of Parkinson's disease. Just want to acknowledge that patients and the carers are really very helpful in the contribution of in the generation of this data. We worked with amazing data scientist Jean Yang's group at the University of Sydney, and also Andy Holmes, who did all of the taxonomy, and just shouting out to my team here who did all of the work in our funding sources. And you'll note here Michael sitting at the other end of me of the table when we're eating dinner. Thanks very much.
Thank you. Yeah, let's leave the question and answer to the end. Yeah. Okay, thank you. So very insightful talk. And that brings us actually to the second talk on the gut barrier that you have mentioned. And it is going to be delivered by Dr. Shanti Dawakala. And she is currently the head of the Gut Barrier and Disease Laboratory in the Department of Anatomy and Physiology at University of Melbourne. And she has done her PhD at Foley Institute of Neuroscience and Mental Health, and where she investigates the role of mitochondria dysfunction in neuronal apoptosis. And subsequently, she has did her postdoctoral training, including that in Uppsala in Sweden. And she has received a Karl Trigger Fellowship to look at memory defects with inhibitor insulin, and also following her return, she has been working with Dr. John Furness, and where she looks at new therapeutics for Parkinson's disease. And today she's going to share with us her findings and also novel insights on gut barrier and Parkinson's disease. So maybe Dr. Shanti, the stage is yours.
Thank you. So there's going to be a little bit of an overlap in the introduction. So I think by the time we finish this session, you'll all be experts in Parkinson's, I think. So I just thought I'd give you an overview of what our lab does. So for the past 3 years or so, our main interest has been how the gut barrier may be involved in various diseases and health states, but we're particularly interested in Parkinson's disease, which luckily fits very well with this session. And so I thought today I'd give you a bit of an overview of what we know about the gut barrier in PD patients, what we see in some animal models of Parkinson's disease, and then show you a little bit of preliminary data that we've generated this year. So just a little overview. Parkinson's is currently one of the fastest growing neurological disorders in the world, but the causes for this aren't really quite known, but ageing seems to be a very important factor, as well as environmental factors and genetic factors, with about 5 to 10% of cases attributed to a single genetic mutation. Overall, the disease affects various regions of the brain, but it predominantly affects the substantia nigra, which gives rise to the loss of dopaminergic neurons and the characteristic motor symptoms such as muscle stiffness and tremors. But Parkinson's patients also suffer from a variety of non-motor symptoms, which include a loss of smell, sleep disorder, and gastrointestinal dysfunctions, just to name a few. And these non-motor and motor symptoms don't often present at the same time and emerge at different stages of disease progression. Unfortunately, though, clinical diagnosis of Parkinson's often occurs when about 50 to 60% of dopaminergic neurons are already lost, making it quite difficult to intervene at this late stage. And this is why a lot of research to date is now focusing on the prodromal symptoms, a stage where there's minimal loss of neurons and a wider therapeutic window for intervention. So gastrointestinal symptoms appear to play a prominent role in this prodromal stage, and this is highlighted by studies that show that constipation can precede the motor symptom onset by more than 20 years. And this link between the gut and the brain the gut and Parkinson's is probably best highlighted by the fact that almost all patients with PD will suffer from some form of gastrointestinal dysfunction. In addition, aggregates of alpha-synuclein, which are thought to drive neuronal loss within the CNS, are also found throughout the gastrointestinal tract. And these aggregates have been observed before motor symptom onset, which has given rise to this idea of maybe PD originates in the gut. And this hypothesis has been studied by various groups and and a lot of labs have shown that in animal models of Parkinson's, aggregated alpha-syn or alpha-synuclein, when it's deposited in the gut, it can propagate to the brain via the vagus nerve and spread disease. And when the vagus is cut, you can abolish the spread of this disease. And interestingly, retrospective clinical studies have also shown that patients who underwent a vagotomy for other health reasons had a reduced risk of developing Parkinson's disease later in life. So again, further suggesting this gut origin of Parkinson's. But as mentioned previously, this gut-to-brain propagation is bidirectional, and it may explain why the pattern of alpha-synuclein aggregate deposition varies among patients. And this has resulted in several different theories of how Parkinson's starts, with some suggesting it exclusively starts in the gut. Some suggest that it exclusively starts in the olfactory bulb and then spreads to other regions. Other theories are that it's a mix of both, the gut and the brain. And then there's another theory where these alpha-synuclein deposits appear sporadically within the brain in populations of neurons that happen to be vulnerable during disease. But regardless of the theory, we think that the intestinal epithelial barrier might play a role in all of these proposed pathways. So very few studies have investigated changes to the intestinal barrier in PD patients, but of the few that have, 2 studies on your guys' left is showing that patients with Parkinson's have small intestinal permeability, whereas some other studies have shown that colon permeability is present in the absence of small intestinal permeability. And other studies have also shown changes in tight junction proteins with reductions in occludin or zonulin occludin 1, and other studies showing increases in alpha-1 antitrypsin and fecal zonulin in stool samples indicative of mucosal damage. So some of these changes in the gut barrier are also mirrored in animal models of disease. So I'm moving away from clinical studies, and now we're really focusing on animal studies, so apologies to clinicians. So this table is a summary of what we know happens in various animal models of Parkinson's. And so we see similar things like increases in intestinal permeability, reductions in tight junction proteins, and changes in goblet cell number and mucin production. And logically, both clinical and animal studies have assessed changes to the intestinal barrier after motor symptom onset or when there's significant damage in the CNS. But we are really interested in understanding what happens to the gut barrier before this time point and whether it could be used as an early marker of disease. So we have been collaborating with a group at the Florey who often let us scavenge gut tissue from their Parkinson's disease animal models. And they routinely use the chemically induced MPTP mouse model, which results in about a 50% reduction in neurons within the substantia nigra and also causes a significant reduction in motor function as assessed here with the POL test. And both of these changes occur 21 days after the last dose of MPTP injection. So we were lucky enough to collect gut tissue from these mice at 7 days, so before any motor symptom onset and before any neuronal loss, and also at 21 days, so when both of these parameters have occurred. So this is still very much a work in progress, but we first wanted to look at gross morphological and structural changes in the mucosa of the ileum and colon, and we performed the standard assessments and measured like villus height, width and muscle thickness in the ileum and crypt depth, width and muscle thickness in the colon. And the earliest changes we saw were in the ileum with a reduction in villus height 7 days after MPTP injection, but the most consistent change that we saw was in the colon with respect to an increase in crypt depth and width, which might suggest crypt hyperplasia, and an increase in epithelial turnover, which is often coinciding with GI inflammation, which we're currently in the process of assessing. But we also saw an increase in muscle thickness within the colon, which may have implications for colon function. And this isn't surprising because this mouse model does show signs of reduced stool output and a reduced water content in pellets, indicative of a constipation phenotype. We next looked at the mucin-producing goblet cells, which also play an important role in maintaining gut barrier function. And interestingly, in the ileum, we saw an early and significant reduction in the number of goblet cells at 7 days when compared to the vehicle, and this persisted out to 21 days. And although we didn't see any change, any significant change in the colon, we did see a shift in the proportion of acid mucins to neutral mucins, so acid in blue and neutral in magenta. But we're currently in the process of trying to analyse this shift and understand how it might relate to disease progression and function in the gastrointestinal tract. We also performed some preliminary assessments on tight junction protein expression levels in the colon using Claudin-1, and we saw a significant reduction in Claudin-1 immunoactivity as early as 7 days, but again, this didn't persist out to 21 days. However, we did notice a change in the distribution of staining within the tissue, which is perhaps indicative of a reorganisation of Claudin-1 within enterocytes. So now I just wanted to move on to another mouse model of Parkinson's that one of our PhD students, Cherish Han, has been working on. And I'll keep this part brief as she's actually presenting in the next session as well. But these mice overexpress the human mutation of the alpha-synuclein gene. And in this, in this model, we don't see any significant loss in neurons within the CNS until after 4 months of age. So much later, like at about 12 months of age. And in our hands, we don't see any motor dysfunction until 6 months or later, and this is assessed using the Ledge beam test. And Cherish has performed a number of in vivo intestinal permeability assays where mice were orally administered a 4 kilodalton fluorescent sugar to assess paracellular transport from the gut to systemic circulation. And using this assay, she's found an increase in intestinal permeability in 3-month-old mice. And this was mirrored in Using data that was generated by Rachel McQuaid, who co-heads the lab as well, a few years prior to what Cherish showed, albeit it wasn't significant. And this is one of the first studies to show a change in gut barrier function at this early time point in this transgenic model of Parkinson's. And I say it's one of the first studies because just last year, late last year, a group in Italy actually showed that these mice at 3 months have a reduction in the tight junction protein zonulin and Claudin-1. However, this doesn't persist out as disease progresses, so this reduction in tight junction protein expression is lost at 9 months. However, they saw a reduction in another tight junction protein, occludin, at 9 months, which suggests maybe there's a transient change in tight junction protein expression as disease progresses. So although we still have many other experiments to do in these models, we have been able to add to this table with respect to changes that occur prior to motor symptom onset, where we've seen in the MPTP mouse model of disease, changes in the ileum with respect to villus atrophy, reductions in goblet cell in the ileum, and also a reduction in tight junction proteins in the colon. And we've also seen early changes in the transgenic model of Parkinson's with an increase in intestinal permeability. So these early changes that we see in some animal models of Parkinson's suggest that we might be able to detect early changes in Parkinson's disease patients as well. So there is some clinical data to suggest that increased intestinal permeability and gastrointestinal inflammation are present in early to mid-stage Parkinson's patients. But again, we don't know what's happening before diagnosis. But other diseases that where intestinal permeability is a prominent feature, like inflammatory bowel disease and irritable bowel syndrome, there is some studies showing that the incidence of getting Parkinson's disease later in life increases if you have a pre-existing gastrointestinal inflammation disorder. But of course, not all patients who have IBD or IBS are going to develop Parkinson's, and this is where other risk factors come into play. So you may remember I mentioned earlier that environmental factors also increase the risk of Parkinson's, and this is another interest of ours. So we're trying to understand whether the combination of increased intestinal permeability and gastrointestinal inflammation and environmental toxin exposure can trigger Parkinson's disease, with the idea being that a toxin gets is ingested and gets into the systemic circulation via a compromised gut barrier to then either enter the blood or gain access to the enteric nervous system to cause disease-modifying changes in the brain, either via the vagus or via systemic circulation. And this could be where the heterogeneity in disease pathology could stem from, as there are various routes these toxins can take once the pollutant enters the body. So Colin Craig, who's another PhD student in the lab, who has performed the majority of these studies, he's trying to model this idea. So he has used orally administered lipopolysaccharide for 7 consecutive days to try and cause GI inflammation and intestinal permeability from within the GI tract. Using this method, he's shown that minimal damage occurs in the ileum, but significant changes occur in the colon. And most importantly, we see an increase in intestinal permeability and an increase in the systemic inflammatory marker C-reactive protein. So using this model, we wanted to investigate if an environmental toxin that is already known to cause Parkinson's disease, disease-like symptoms and pathology in mice, could cause PD-like pathology or symptoms when given at a dose that isn't normally associated with disease onset. So the pesticide we chose was rotenone, which is a mitochondrial complex 1 inhibitor that has been widely used to induce animal models of Parkinson's. And I'm not expecting anyone to be able to read this table, and I've just purely put it up just to show that many people and many groups have used this toxin to induce Parkinson's disease-like symptoms and pathology in animals, and to highlight that when these groups do use this toxin orally, they need to use doses of 30 milligrams per kilogram or higher to induce both motor and non-motor symptoms or CNS damage. So for our experiment, we wanted to see if a shorter exposure to rotenone would induce PD-like symptoms if the gut barrier was compromised. So in order to do this, the experiment involved having several groups that either received the LPS vehicle or LPS for 7 days, but halfway through that treatment, the appropriate cohorts received rotenone or the rotenone vehicle for an additional 7 days. Various tests for motor and gastrointestinal function were then performed 1 week after the last dose of rotenone, and then every 4 weeks thereafter. Because we didn't know what effect this toxin or this combination was going to have on the mice, the best way we could monitor them, other than visually, was to weigh them daily throughout the dosing period, which is the graph on the left. We didn't see much change in body weight between the groups until mice started receiving both LPS and rotenone, which is indicated by the red arrow and the grey line. And as you can see, as soon as we started administering both in combination, we got an immediate drop in body weight. And this change in body weight was sustained throughout the dosing period and was significant when compared to the LPS vehicle mice. And we continued to monitor these mice over a period of 3 months, and we're still continuing to monitor them as the experiment hasn't finished yet, but we still see this reduction in weight. So these mice aren't gaining as much weight as all the other groups, which suggests that this combination of toxin and leaky gut does have some effect at the level of the GI tract. And given that constipation is frequently observed GI symptom, we wanted to assess a potential constipation phenotype in our model. So we did this using the bead expulsion test, which involves inserting a 3 millimetre bead 2 centimetres into the colon and recording the time for the bead to be expelled. We still have a few time points to assess, but we are starting to see an increase in bead expulsion at 16 weeks, which is indicative of reduced colon motility in all of the groups when compared to the LPS vehicle group. So it should be interesting to see what happens as we collect more data and add more animals to this graph. We are very aware that the variability within this test is huge. Yes, but as I said, we're going to add more animals to these cohorts, and hopefully that variability will reduce and we'll start seeing some significant differences. We've also continued to assess intestinal permeability in these mice. So 1 week after finishing dosing of the toxin, the increase in intestinal ability was maintained in the LPS group, and we did see an increased trend in the LPS and rotenone group, but this dropped off as we kept monitoring the mice. And this wasn't surprising given that the mucosal barrier is probably renewing and turning over the cells in the mucosa, but it does seem to fluctuate from month to month. We've also been assessing motor function using the Ledge Bean test, which predominantly assesses hind limb function and coordination. And although nothing is significant yet, we are seeing an increasing trend towards a motor deficit in the LPS and rotenone mice, which is shown in the grey bar here. And on the left, we're looking at the total number of foot faults they produce on the Ledge Bean. And on the right, we've got this the time it takes for them to cross the beam, which is also indicative of motor function. And yes, we're seeing also an increase in the LPS and rotenone group again. Another test that we've used to assess motor function is the Rotarod test, which measures balance and coordination. Again, we're seeing a motor deficit appearing that hasn't quite reached significance yet, with mice in the LPS rotenone group falling off the Rotarod apparatus quicker, and obviously making a greater total number of falls. But we've still got 2 to 3 months left of behaviour to perform, so we anticipate these trends will continue. So as I've mentioned a number of times, we still have many more experiments to do, and our next step is to really focus on what's happening at the cellular level in both the gut and the brain. But from the preliminary data we've collected so far, we do see early changes in the gut barrier in both the upper and lower GI tract with respect to morphology, intestinal permeability, and tight junction protein expression. And we're starting to see evidence that that the gut barrier when the gut barrier is compromised, that it may increase the susceptibility to developing Parkinson's when mice are exposed to doses of toxin that would not normally cause Parkinson's-like symptoms. So we think if intestinal barrier dysfunction does contribute to disease onset or even progression, could protecting the gut barrier alleviate symptoms, slow disease progression, or better yet, completely stop disease spread. So just finish off by thanking everyone in the lab, and yeah, of course, John's lab, who often let us use some of their space and reagents, and our collaborators. Thanks.
Thanks very much. Michael Szczesnak is now going to come and present to us. He's a scientist with real expertise in upper esophageal motility issues, and he's going to talk about these issues and Parkinson's disease. Michael.
Thank you very much for the introduction. Welcome, everyone, and it's a great pleasure to share our findings with you today. And I'll talk about oropharyngeal dysphagia in Parkinson's disease, but more importantly, what we can potentially do to treat it. And I will not sing, it's a scary thought. So Parkinson's disease after Alzheimer's disease is the second most common neurodegenerative disorder. And oropharyngeal dysphagia in Parkinson's disease is very common. Almost all patients end up with dysphagia in the course of their disease. And it not only impacts on the quality of life, but also on survival. These patients have increased risk of aspiration pneumonia, and mortality after first occurrence of aspiration pneumonia is 24% at 1 month, 65% at 1 year, and 92% at 5 years. So I don't like those odds. And what's also disappointing is dysphagia is largely unresponsive to pharmacological treatment. Now, that's nearly 30 years ago. Our mentor, Professor Ian Cook, and his team, using old-school water-perfused manometry and sleeve sensors, have shown that upper esophageal sphincter dysfunction is an important pathophysiology in Parkinson's disease. But before I get any further, let's just establish what the upper esophageal sphincter is. When we talk about upper esophageal sphincter, we're looking at the junction between the pharynx and the esophagus, and it's a high-pressure zone, major component of which is the cricopharyngeus muscle. And it's quite a peculiar muscle, and it's C-shaped, and it attaches on the back of the cricoid plate, and it contributes to most of the pressure generated by the sphincter. Now, upper esophageal sphincter is composed of skeletal muscle that's under central nervous system control, and at rest, closure of the sphincter is due to both passive forces as well as active muscle contraction, and its relaxation requires inhibition of the cricopharyngeus. And this is very nicely illustrated by the EMG studies of the cricopharyngeus. On the top, you can see a normal subjects, and at rest, there's an active tone in the cricopharyngeus, and during a swallow, there's an inhibition that allows this sphincter to relax and swallow to occur. On the bottom, we have a Parkinson's disease patient, and you can see there is absence of that quiescence period in the CP muscle, resulting in failed relaxation. Now, that's not the only problem, and it doesn't happen in every patient with dysphagia. There are other mechanisms of oropharyngeal dysphagia in Parkinson's disease, like poor lingual control, delayed swallow initiation. Now, can we learn more about it using more modern technology, or will we be just reinventing a circle because we have new toys to play with? And more importantly, can we treat it? Now, the results I'll present today were done by Peter Wu on his fellowship in Hong Kong at Prince of Wales Hospital. And I have to say, this was done during a period of civil unrest. He had to dodge Molotov cocktails and tear gas to get this done, so a lot of effort went into this. Now, where I come into this, I was in Sydney doing all the number crunching and analysis. That's why I get to present this. So we studied 64 consecutive patients with Parkinson's disease. These were patients with or without dysphagia, and we compared them to age-matched healthy controls from our existing database. So swallow function was assessed with Sydney Swallow Questionnaire. Dysphagia severity was assessed with Sydney Swallow Questionnaire. The Parkinson's disease severity was assessed using a validated scale by a neurologist, and quality of life was assessed with SWAL-QOL, or Swallow-Related Quality of Life Questionnaire. And all patients underwent pharyngeal high-resolution impedance manometry. I'll cover it just very briefly because you'll see a lot of it in the later sessions. So patients were intubated with a pressure and impedance catheter. We performed a number of swallows, generated spatiotemporal images, and analysis was performed on swallow gateway. But the variables of interest that we looked at was the upper esophageal sphincter relaxation pressure, which is the nadir pressure during swallow, obviously. UOS relaxation time, so it's just the duration of relaxation. maximum admittance, which is a marker of the maximum opening diameter of the upper esophageal sphincter. Intrabolus pressure, which is pressure above the sphincter as bolus flows through it, and it's a marker of flow resistance that the bolus encounters while it's being propelled into the esophagus. And pharyngeal contractile integral, which is simply a measure of pharyngeal propulsion. Our patients were well matched in terms of age and gender. And it's important to note the SSQ score here is quite modest. It goes all the way up to 1700, and only 50% of patients were actually dysphagic. And again, on average, the Parkinson's disease severity was moderate. Now, what we found that dysphagia severity was associated with Parkinson's disease severity. Not a very strong association, but what I would like to point out, is a pointer, if you look at the patients with mild disease severity or Parkinson's disease severity on the x-axis, they still can have quite significant dysphagia symptoms. And as the Parkinson's disease severity gets worse, they all end up with dysphagia, except that one outlier at the end. Now, so what we found with manometry, so here we have upper esophageal relaxation pressure for different bolus volume that we tested. There is a volume effect, which we expect, but we found that the upper esophageal sphincter relaxation pressure was elevated in these patients. The relaxation time was reduced, so the sphincter was relaxing for a shorter period of time, so there was less time for flow to occur. Maximum admittance, or a marker of opening diameter, remained the same, or there was no difference between the groups. The intrabolus pressure was elevated, indicating increased resistance to flow. And what we found interesting is that the pharyngeal propulsion was elevated as well, which we think is a compensatory mechanism for the increased resistance to flow. And here's just a couple of example traces from healthy control on the left and Parkinson's disease on the right. On the left, the sphincter zone is that hot band right in the middle, and the purple areas on the bottom indicate where flow is occurring. And you can see that during the period of flow, the sphincter is nice and open and fully relaxed. But in the patient, the sphincter relaxes, there's flow occurring, but very briefly, and there's still a lot of residual pressure and upstream pressure as well, so high intrabolus pressure above the sphincter zone. And these two markers, intrabolus pressure and US relaxation pressure, also correlated with Parkinson's disease severity. So I think it is quite clear that at least some of these patients have restrictive upper esophageal sphincter abnormalities. They have increased pharyngeal contractile pressures, a possible compensation for increased resistance at the upper esophageal sphincter. And Parkinson's disease severity was positively associated with dysphagia symptoms and elevated intrabolus pressure and US relaxation pressure. Now, but what's more important, the most important finding here is that they have restrictive upper esophageal sphincter abnormalities. So what can we do about the problem? Well, we're good at breaking things, so why not just cut the offending muscle? And transcutaneous cricopharyngeal myotomy has been around for a while. It works in 60 to 70% of cases. However, it is associated with unacceptably high mortality, morbidity with quite serious side effects, so it's rarely performed. So it kind of works, but it's really not good enough. Can we do this endoscopically? Well, it just happens that peroral endoscopic myotomy, so myotomy of the lower esophageal sphincter, has been around for over a decade now. It's accepted treatment, works well. So why can't we apply this technique to the sphincter on the other end of the esophagus? And so Peter Wu developed this technique while in doing the fellowship in Hong Kong. First testing it on pigs and then applying it to intact humans. So I'll just walk you through this briefly. So The principles is same as standard poem, it's just higher up. And the entry point is in the posterior hypopharynx, just opposite to area epiglottic folds, and injection is made into the submucosa to create a cushion. And this is followed by 1.5 centimeter mucosotomy, and it needs to be done carefully to avoid damage to the neighboring laryngeal structures. Then the submucosal tunnel is created past the CP muscle to expose the muscle fibers, and you can see these horizontal bands on panel B, and the tunnel length is about 5 to 6 centimeters. Then myotomy is performed of a total length of 3 to 4 centimeters, and you can see the completed myotomy in panel C with exposed posterior adventitia, and then the mucosal defect is closed with series of mucosal clips at the end of the procedure. Now, out of those 64 patients that we studied, half had dysphagia, and of those, 11 had demonstrable failed upper esophageal sphincter relaxation on impedance manometry. Now, CPAP was performed in 8 of these patients. 1 declined, 2 were excluded, 1 because of high anesthetic risk, and the other patient had deep brain stimulator, which is a contraindication for monopolar diathermy. The myotomy was achieved in 100% of cases, and the procedure time was around 66 minutes. Surgical emphysema was present in all cases. This is to be expected. However, there were no ventilatory issues. Patients were able to resume medication immediately after recovery. And most importantly, there were no perforations, infections, or delayed bleeding. And this is something we worried about, but the hypopharyngeal clips were well tolerated and minimal analgesia was requirement. There was minimal analgesia requirement. And here's an example of a manometric trace before CPOM and after CPOM on the right, and you can see the UES time has been completely abolished, but most importantly, the pharyngeal contraction is well preserved, indicating that myomedy did not significantly extend into pharyngeal constrictures, which is something we don't want. Okay, so how did the patient's swallowing go? So here are the Sydney Swallow questionnaire scores before and after CPAM, and there was an improvement. But to many of you, I'm sure it's just a number. So I will just put this into a bit of perspective. So the dotted line is upper limit of normal. Once you start getting to sort of scores of 500, you really have to avoid the troublesome foods like bread, fruit, hard fruit, steak. Once you get into 1,000, you're really limited to a single consistency, and maintaining weight becomes an issue. The upper limit is 1,700, and in that territory, you really need a PEG tube to maintain nutrition and hydration. And we did see significant improvement in dysphagia severity in these patients. And unsurprisingly, it also translated to improved swallow-related quality of life. Now, the US metrics also improved, so integrated relaxation, the US relaxation pressure decreased in many cases, going into the normal range, which is shaded in gray. Intrabolus pressure also decreased. Maximum admittance was not changed, however, it was normal to begin with in many cases. Now, relaxation time did not change, but if you look closely at those dots, the ones that were normal to begin with remain normal. The ones that were reduced normalized, but overall there was no significant difference. Now, if that's not convincing, we also performed video fluoroscopic studies on these patients, and we measured the cross-sectional diameter of the upper esophageal sphincter during a single frame by frame during a swallow of a 10 ml thin barium suspension, and this will be mapped as an opening diameter over time, and this gives us a relaxation time, maximum opening diameter, and by calculating the area under the curve, we get a good measurement of both of these aspects. So on the bottom of the axis, we have the baseline measures. And above the x-axis, we have the post-CPOM measures for each patient, and you can see that the maximum diameter has increased, the duration of relaxation has increased, and also the area under the curve is elevated. Now, all of these measures were taken 1 month after CPOM. And we followed these patients out to 9 months just to see how robust the treatment is and whether it's maintained over time. And it looks like the response is maintained at least out to 9 months. So CPOM is feasible and appears safe. It induces objectively measured improvement in upper esophageal sphincter relaxation and flow resistance. It induces improvement in swallowing function and swallow-related quality of life. And it's obviously premature to draw any conclusions, but this preliminary data is promising. And there are still some key questions to be answered. How to select who will benefit the most from CPOM, and I think manometry will be great help here. And also, to what extent can CPOM improve oropharyngeal dysphagia and Parkinson's disease in real-world scenarios and also in other myogenic disorders? And we are currently undertaking a multi-center randomized control trial comparing CPOM versus swallow therapy delivered by speech pathologist. But I won't get into this. Hopefully, in a couple of years' time, we can present these results. Now, failed upper esophageal sphincter relaxation is not isolated to Parkinson's disease. It's also an underlying mechanism in many other neuromyogenic dysphagia disorders. And this is a local experience at St. George. And Peter has done CPOM in lateral medullary stroke, a couple of Parkinson's patients, myositis, head and neck cancers, and patient after tumor resection. And the response is comparable. And what's really encouraging is that some of these patients, at the top of the scale, dropped down to near normal diet, and they were able, after 10 years or how many years on PEG tubes, to have it removed and resume a near normal diet. So this is really encouraging. Now we have POEM, GPOEM, ZPOEM. Why do we need another CPOEM? Now, POEM is for achalasia, is performed routinely, and it is a very uncommon disease with prevalence of 3 to 4 per 100,000 in population. Now, in contrast, there are many neuromyogenic diseases, and in Parkinson's disease alone, there's estimates of global prevalence of 9 million by 2030. And most of these patients will have dysphagia. And combining this with other neuromyogenic causes, there's a great potential for CPOM to improve quality of life in these patients and improve survival. Okay, that's it. Thank you.
Thank you, Michael, and has been a great section so far. After the first talk, I feel I'm walking a bit slower. After the second one, I felt my tummy a bit wonky, and after the first speaker, I feel a bit dysphagic. And, so that brings us to the fourth speaker, so Ms. Cherish Han, and she graduated from University of Melbourne in 2019, and also did her studies honors, and also currently PhD study with Dr. Shanti, and that she is going to explain my GI symptoms and also talk about GI symptoms in Parkinson's disease. Ms. Han, please.
Thank you so much for introducing me, and I'm very thrilled to be here. But first, a mini apology, because I just realized me and Dr. Caroline Su, we start this presentation the exact same way. But this is a very influential paper by Dr. James Parkinson. So yesterday, Professor Nick Talley talked about a paper from 1910 This actually goes back to 1817, 200 years ago. And yes, when James Parkinson wrote about documented Parkinson's disease, he also noted that a lot of his patients were also experiencing a lot of the GI problems, such as dysphagia, constipation, dyspepsia, and nausea. And since then, we've come to really appreciate the GI symptoms in Parkinson's as evidenced by this very symposium. And we also know that they affect up to 90% of patients at least at some points in the course of the disease, and more often than not, they precede the onset of motor symptoms by many years. So Dr. Caroline touched on the alpha-synuclein a little bit. I want to talk a little bit more about the protein later on as well. But first, PD is notoriously an old age disease that usually manifests after age of 60. But there's a subset of PD called a young onset PD with an early age of onset. And this type of PDs has even a higher prevalence of GI symptoms, which affects up to 95% of patients. So about alpha-synuclein. So one of the main, Parkinson's is obviously a very complex disease, but one of the main pathological hallmarks of PD is the abnormal accumulation of alpha-synuclein in the brain. And they form this very distinct pathology called Lewy bodies or Lewy neurites. And recently there's increasing evidence suggesting that this alpha-synuclein buildup actually extends beyond the brain and reaches the enteric nervous system as well. So what is this protein? So alpha-synuclein is a very small protein located in the presynaptic terminals of the brain and also various parts of the nervous system. And it has a very important central and diverse role in regulating the synaptic transmission by interacting with multiple molecular targets. So I have here a very simplified diagram outlining some of the functions, which includes maintaining the cytoskeletal structures, influencing the mitochondrial fiction and fusion dynamics, and facilitating endocytosis, modulating the dopamine transporter activity, and also participating in activity-dependent exocytosis. Additionally, the healthy form of alpha-syn can also modify the membrane permeability by incorporating itself into the cell membrane to create ion channels. So obviously, this very important protein, it exists in the presynaptic terminal, but sometimes there are some triggers that cause this protein to aggregate. And when that happens, all of these physiological functions, they become disturbed. It can destabilize the intracellular cytoskeletal structures, which disrupts the mitochondrial fiction dynamics and hinders membrane localization of the dopamine transporters. It also impairs endocytosis. And aggregated ASIN can also be released into the surroundings through exocytosis. And for the last point, I'm going to come back to it in a later point in the presentation. So earlier I mentioned that the young onset PD is often inherited. And turns out the mutations in the alpha-synuclein gene is actually involved in a lot of the familiar forms of PD. So, and one of the mutations is called A53T. It's a point mutation in the N-terminal region of the alpha-synuclein protein, and it results in accelerated fibrillation of this protein. And that fibrillation is accelerated because of the beta-pleated sheet formation. And the A53 transgenic mice we're going to use in my project, it has an overexpression of the human A53 variant of the gene, and it's driven by the mouse prior protein promoter. And using this model, we're going to assess the gastrointestinal deficits during the early stages of the disease progression and also the underlying neuropathology. So in my study, there are 8 cohorts of the wild type and its transgenic mice, and we assess like behavior, and we call them at 7 weeks intervals from 12 to 36 weeks. And the oldest from these mice will probably correspond to about middle age in human years. And each time point, we use various experimental methods to track the progression of central motor deficits and, of course, gut motility dysfunction alongside alterations in the ENS. First, we looked at the central motor deficits. in the model with the ledge beam test. So this is a test that has where we use a long, narrow beam. It's a bit wider at the other end, and the other end is connected to the home cage. So we train the mouse to go from the wider end to the narrower end, and the beam also has a ledge on the side, which serves like a safety net for the mice. So in the actual trial, we record the amount of foot slips when it was going from one end to another, and that's like a measure for motor movement, for movement and coordination balance. So we found there are pretty profound changes there's a clear separation between the wild type and the FD3 mice, which is expected because this mouse model it usually has a very progressive phenotype, a really aggressive motor phenotype. And it starts pretty early from like 24 weeks of age. So next, we are going to look at motility, because we're a motility conference. And we used the whole gut transit test first. So in this test, we gavage the mice with a non-absorbable dye, and then we record the amount of time it takes for the mice to pass the dye in the stool. And that serves as a measure for whole gut motility. Sorry, I went too fast. There you go. So in the whole gut transit test, we did not see any difference between the two genotypes. So it turns out that the amount of time it transits, the amount of transit time like for the whole gut, it actually doesn't really change that much in these mice. So next, we want to look at a specific section of the gut. We chose the colon. And to look at it, we use the bead expulsion test. So there's a very small bead that we use, and we put it into the rectum of the mice, about 2 centimeters up into the rectum, and we again record the time it takes for the mice to expel it. The bead is not it's true to scale. It's actually much tinier. And we saw a pretty remarkable change from very early age at 16 weeks. So these mice, they have slower colony motility. So we want to find out what's going on in the enteric nervous system. So we looked at the cellular morphology. To do this, we counted the number of neurons in the enteric nervous system. We used the hue marker, which is a pan-neuronal marker, so that we can look at all the neurons in the ENS. We did not see any significant difference at any of the time points, but there's a trend for the FD3 mice to have a lower number of cells, and it also has a higher loss of neurons over time when we compare the 12 weeks time points to the 6-week time point. So next, we also looked at the nNOS positive neurons, which, as many of us are familiar, which are inhibitory neurons, inhibitory motor neurons, and they're also known to be vulnerable to oxidative stress. Again, there wasn't any observable difference between the two genotypes, and there's an age-related decrease as well. So when I was counting the neurons, I used this program called the Gut Analysis Toolbox, and that program actually outlines the cell area of all the cells that I counted. So I was like, we can also look at the cell size. And it's pretty interesting that in most of the time point, the FD3 mice actually has a bigger cell size compared to the wild type, but the difference is not very significant except at some of the time points, but I think it's a very interesting parameter to look at. So we have slow colony motility in the mice with no change in anterior neuronal number size. So what is going on in the ENS? So we turned our attention to the glial cells. I know that there are a lot of people in the audience who are fans of glial cells, but for those who are not, so the largest collection of glia outside of the brain and spinal cord is housed within the ENS. And enteric glia are one of the most dynamic cell types in the gastrointestinal tract, and they have a lot of diverse roles in neuronal support, mucosal integrity, neuroprotection, neurogenesis, and also neuroimmune interactions, and most importantly, synaptic transmission. And there's increasing evidence for the nuclear interactions. Increased evidence that nuclear interactions are very important for the ENS functions. And because neuropathology can be broadly defined as like a failure of the nervous system to maintain local homeostasis, I think it's really important that we're also looking at glial cells when we're looking at neuropathology in many of these diseases, including Parkinson's. So we counted the number of glial cells labeled with two glial markers, S1 beta and SOX10, both of which show profound changes at a very early age. So I forgot to mention that all of these cellular studies, they're all done in the myotory plexus. So we found significant changes in the colon, which is in line with what we saw in the motility test, slow colonic motility. And actually, loss of enteroglial cells has been correlated with chronic constipation in literature, but it hasn't been looked at in like any of the PD models, especially in the mouse models. So this is the first like pretty novel results. But there's also like an age-related decline in the glial cells, but very interestingly, it doesn't, the differential expression is not maintained in the later course of the disease. It only shows up in the very early age. So back to the neurons. There's no morphology changes, so what about the functional changes? So to look at it, we used calcium imaging. So live calcium imaging is it's a very common technique, but it hasn't been used much in the animal models in PD, as far as I know. And it has a very decent physiological relevance because it keeps things in a relatively intact natural environment where the synaptic pathways and cell-to-cell interactions are all preserved. So again, this is the myotory plexus preparation with the cellular muscle still intact. I loaded them with the fluorophore AM dye and imaged at 120 frames per minute. And I used two solutions to induce action potentials, high potassium solution and cholinergic receptor agonist DMPP. So we found some remarkable changes in intracellular calcium signaling in the AFT30 mice. There's an increase in intracellular calcium concentration like transient. This is calculated from an average change from baseline to peak. And we saw this like a similar phenomenon where the change is not preserved in older mice. It actually reversed. We also saw the similar results when we did with DMPP. We did calcium imaging with DMPP and increased intracellular calcium signaling. So what's going on in these very early age mice? So I want to come back to this slide again to talk about more of the pathological effects of aggregated ASIN. As I mentioned, alpha-syn affects calcium, affects membrane permeability by interacting with it and forming ion channels. And turns out these channels are actually permeable to calcium and cause changes in calcium concentrations. And so when there's aggregation in the alpha-syn, especially the type with lots of beta sheet structures, which is what happens in the AFT30 variant, the membrane permeabilization results in higher calcium spikes. And it's thought that this is because the pores that they're forming are actually more stable when there's these aggregated beta sheet structures. And also, in addition to calcium dysregulation, these aggregated oligomers, they also release reactive oxygen species, which exacerbate cytotoxicity, and eventually leads to cell death. And from the results that I just showed, these changes are happening pretty early on in the ENS. So the relationship between alpha-syn aggregation and calcium dysregulation, it's actually been studied in the CNS for a while in different animal models, but it hasn't been looked at in the ENS much. So as we know, there are a lot of different mechanisms at play, one of them, which I just mentioned, forming the calcium channel pores. There's also another study that shows that alterations in the mitochondria associated with increased intracellular calcium levels. It was shown in a double mutation model in 2 PD familiar genes, one of which is the A53T, another one is the pink one gene. There's also growing evidence that calcium also promotes alpha-synuclein aggregation, both directly and indirectly, which again results into this positive feedback loop between calcium dysregulation and alpha-synuclein aggregation. So we think that the gut dysfunction in the A53 model of the mice, it might be associated with differential calcium signaling and enteric glial loss, which might be mediated by the synaptic dysfunction caused by the aggregation of alpha-synuclein. So the exact role of enteric glial cells in the pathophysiology still needs to be investigated, but understanding these mechanisms will be very invaluable for future therapy. Thank you so much. I still have one more slide. I'd like to thank everyone who's been teaching me and training me since my honors, especially people from Finance Lab, and also especially my supervisor, Shanti and Rachel, and also especially Marlene for letting me use the calcium imaging rig. And also I would like to extend my thanks to Dr. Peter Vanderberg, who lets me use the analysis program for calcium imaging results. Thank you so much.
If I could call the panel up, or all the speakers up, please, to the panel discussion. We have a few minutes now, about 15, I hope, for questions, if there are questions from the audience, which I hope there will be. That was a very stimulating session, a very interesting gut-brain disease discussion, and there's an awful lot we don't know. So let me ask Carolyn first of all. So Carolyn, you showed there are some microbiome changes in Parkinson's versus the household controls, which were really quite interesting, and there were big differences in some of those different bacteria genre, etc. But they were also, the Parkinson's patients are also largely constipated. So what's chicken and what's egg? Is it that constipation is the primary problem in Parkinson's and you get microbiome changes that follow? Or is it possible the microbiome changes from Parkinson's induce the constipation, and indeed that's a primary driver of the disease process? So which comes first and how do you work it out? Because it seems remarkably important, if we're going to intervene, to actually understand that particular question.
Thanks, Nick. It's a really good question, obviously. And when it can be one or t'other, it's probably both, or a little bit of both. What we do know, nobody knows this, of course, but what we do know is that patients who are constipated, and this is obviously a non-specific symptom, don't all get Parkinson's disease. And then also, one other study that I'll just quote to you, which actually started me on this journey of understanding the changes of microbiome in Parkinson's disease patient, was a Cell paper by Sampson and colleagues who basically fed fecal samples from patients with Parkinson's disease and patients controls to the A53T model, alpha-synuclein transgenic model, and those mice that were fed the Parkinson's disease poo, as I'll call it, actually developed Parkinson's disease faster than not. So we know that the microbiome, presumably from those fecal samples, could induce and accelerate the disease progression, at least in the mice. So you can't do that experiment in humans, obviously, but nevertheless, you can try. What we're going to try and do is to modulate that microbiome to see if we can impact and modulate or reduce disease progression, and then hopefully we'll know.
Yep. Thank you. Let's see if there's questions from the audience. I've got plenty of questions. Please, down here.
Really excellent talks. Thanks so much.
Lincoln, you don't expect our mics to work here, do you?
Hello, yes, okay. This is actually going to be an annoying question because I'm going to ask it to anyone on the panel and I'm going to ask you to speculate, I guess. So there's people who are interested in companies that are now going into clinical trials for stem cell therapies to replace the dopamine Parkinson's disease patients. And I'm going to ask the panel or someone to speculate on what you think might happen to the gastrointestinal manifestations when you restore the dopamine lost, and if you think that the gut might still pose a threat to those newly made dopaminergic.
That's a tough question.
Well, yes, given that we've shown this bidirectional crosstalk between the gut and brain, probably, and Cherish has also shown that enteric glial cells also play an important role in potentially colon motility, even though if you do stem cell therapy within the brain, you probably may have these gut dysfunction manifestations. And this whole idea that maybe alpha-syn can propagate from the gut to the brain, if it's still in the gut, there's still potential for it to go to the brain again and cause disease. And they've already shown, in some animal models of where you've injected stem cells and ACN aggregates that are already present in the brain can migrate to these newly injected neurons and cause disease again. So potentially may not have an effect, but I don't want to dampen any of those hopes with the stem cell therapy, but yeah. If anyone else wants.
But it is an issue though, isn't it? Because the vagotomy story is very interesting with Parkinson's disease that you pointed out, or one of you pointed out in your talk, and it's really struck me, that particular clinical data, that if you've had a vagotomy, you're somewhat protected, or at least it seems that way. And you just wonder if you're going to put stuff in the brain, whether you should be thinking about blocking the gut off as well. And maybe you'll need to do some kind of vagotomy-like approach. I'm not sure what that would be, but it really is an interesting thought.
Yeah.
Carolyn?
Yeah, no, look, I think, Parkinson's disease, is a multi-system disease, as you say. So one thing to think about is that we know from the stem cell transplant studies that have been done, both back in the 1990s, 2000s, and also the ones coming through, that if you put in, and that's in the early days, fetal transplants, fetal stem cell transplants, then there were post-mortem studies with those patients who showed that there was an accumulation of alpha-synuclein in those fetal transplants. And so this is where the concept of alpha-synuclein propagation and transmission came about. So I suspect that, let's assume that alpha-synuclein is also entering in through the gastrointestinal tract, plus also being transmitted or propagated from neurone to neurone, then the stem cell transplants will accumulate the alpha-synuclein, the degenerative process, which again feeds forward into a mechanism of towards neurodegeneration. So I suspect that regardless of what we do in the head, you guys are going to have a great contribution to make as we try and reduce this pathway by which you can modulate and you can reduce the accumulation of alpha-synuclein through the gastrointestinal tract. And if you think about alpha-synuclein, it's like if you put alpha-synuclein in a beaker, it just sits there. But once you spike it with abnormal or aggregated protofibrils, then it all sort of gloops up. It's a bit like an acrylamide gel for those people who are old enough like me, to sort of put the TMED into our acrylamide gels, and then it all sort of polymerizes and goops up. That's the same as what happens in alpha-synuclein. So the idea is to reduce that, and so modulation of the microbiome, I think, will play an important role in the treatment of Parkinson's disease and its progression.
Other questions, please. Up the back, yeah.
Can you hear me okay? Yeah. So the last two speakers, Maya and Shanti, why you use the bead pellet expulsion? Why do you do that?
Oh, it's just one method. Yeah, it's just one method that we use to measure colon motility.
But you didn't show any other methods, right?
No, but we do also do whole gut transit to look at entire gut transit, but obviously that doesn't just look at colon motility. And we have in the past done spatiotemporal mapping of the colon ex vivo. Yeah.
No, I know all that, that's great. But it seems to me you're probably missing the best and simplest experiment, and that is just count the pellets.
Oh, sorry. Yep, we do fecal pellet output as well. So counting pellets. So we haven't. So in our models, we haven't seen any change in the A53T. In our LPS model, we're just starting to see a reduction. It's not significant again, but I'm hoping as we progress in our study, we might see that reduction in stool output as well.
And just a comment, there's a lot of the talks at this meeting and a lot of the other meetings, there's rodents are used as models. But I can tell you that you can knock out half the ENS and you might not see much change in gains. We published that about 20 years ago. So don't be upset if you don't see changes. It looks like you're not seeing big changes, and that's what I'd expect because the ENS, there's so many more neurons there than you think. It's evolved over millions of years like a drum. If you think of Parkinson's, you're losing 90% of substantia nigra before you see any phenotype. So the vast majority of the cells are gone. And that's the same with the ENS from that. So 5 or 10%, 20, even 30, 40%. I'll be shocked if you see any difference. I've just published the science and then let the field sort of evolve with it, don't sort of hunt around for results.
Thanks, Nick. More questions, please. Yep, Emad.
I'll just screen. Presentations, all four presentations. Sounds like with the animal models, you have a unique opportunity to sort out the chicken and the egg, especially if you start from the beginning, in other words, before the manifestations of the disease, and monitor all these factors, microbiome, all the usual things in the model, and intervene. In other words, if you're able to reverse those microbiome changes and see whether that affects the phenotype, how quickly it's expressed, quickly it progresses. Has that been done? I mean, is this something that you're incorporating in your studies? Because if you have a unique opportunity with a longitudinal design in a murine model, which you can do in humans, but just proof of principle.
I mean, we would love to do some microbiome analysis in our models. So we've been collecting pellets throughout the whole study in the hopes that we will do that. So we've done it before, we've given the treatments and all the way during. A lot of people have done microbiome analysis in animal models of Parkinson's as well and have shown shifts in microbiome. Again, difficult to say what those shifts mean and what's driving, what microbiome changes are driving any change in GI function as well or CNS function. But they've definitely been done. And
before the expression of the disease, though.
If
what I mean is that you can reverse it. You try and reverse it by changing the microbiome or something, so that you can prove that in fact, the chicken and the egg thing. In other words, if it's going to be a microbiome-related issue, you do pre-morbid before the disease is actually expressed. Because it's a longitudinal model. You give them a dive or an FMT or something that actually
stop the disease.
Reverses those abnormalities of the microbiome.
So we're hoping with our study, if we can intervene early, so the next step was going to be to give it some sort of intervention before we see any change and see if we could either stop it or reverse the effects or, But definitely.
Yeah, just to chip in, I was involved in the study back in Hong Kong that we look at RBD, the REM sleep disorder, which is kind of a established prodrome of Parkinson's disease, and we saw some changes over there. So I'm going to share that tomorrow morning. So I think that's quite interesting. We haven't really done the causality moving into animal, which actually I think would be a great study. But I think we saw some early changes in the microbiome, so that might support in that as well.
Thank you. More questions, please. Right at the back there, because I think you've been waiting for a bit. Hand up. Yep.
Question for Dr. Michael, Anson, North Shore Hospital. So when you do a myotomy for achalasia at the lower esophageal sphincter, there can be complications, and I was really interested. As expected, you do this seed foam, and you can
For example, like regurgitation or reflux ways. We haven't observed any changes or any such events so far, but then again, we haven't really closely followed these patients. We haven't done reflux studies on them afterwards. But it is something we should really look into to make sure they don't have aspiration of refluxate instead of food or liquids.
Thank you. Question down here.
Thanks for the great talk. Sally Single from RMIT Melbourne. And I wanted for Shanti, just on the permeability, I was interested to see that you're looking at the ileum and the colon, and we have also looked in those areas. And we recently found that actually we've seen a really stark change in the duodenum, and I'm just wondering, have you looked higher up?
So we have collected tissue from the duodenum, and once we finish this study, we'll also collect tissue from the duodenum, because I think most of our changes are actually in the duodenum. But we're in the process of measuring all that data and assessing it. But yeah, definitely we're looking at higher up in the GI tract.
Everything happens in the duodenum. For those of us interested in the duodenum. More questions, please. Are there any more hands? Yes.
One comment, because one of the clinical syndromes that is associated with future development of Parkinson's is gastroparesis. Gastroparesis, not functional dyspepsia though, but gastroparesis is actually one of them. So I think probably duodenum and stomach might be quite relevant in terms of
Just to add to that, so we did do some post-mortem studies on those mice that we gave the dual treatment, and we noticed, their stomach was full of contents and like huge, so quite distended. So it does look like there is some sort of gastroparesis involved in that dual combination of the toxin and LPS.
There was actually one mouse model where they inject fibrin into the GI tissue, and I was told that actually they inject into the duodenum. So, but that took probably a year to develop. So I don't know how established it is.
Any final questions or comments? If not, we're going to wrap it up. Thank you very much, everybody. Brilliant. Thank you, Sunny, for co-chairing, and have a great lunch.